Welcome learners. Today in this session you will be studying about the biomolecules, proteins. Proteins are large macromolecules consisting of one or more chains of amino acid residues. They are in fact made up of building blocks known as the amino acids. They are complex and abundantly found in all the living systems. They differ from the carbohydrates as they contain nitrogen and in some cases the sulfur in addition to the carbon, hydrogen and the oxygen. The proteins contain a carboxylic group and an amino group. The carboxylic group, as you know, is the COOH and the amino group NH2. Hence, they are also referred to as the Zwitter ions. There are about 21 amino acids that form the proteins. The amino acids have been categorized or classified based on number 1, the structure, number 2, the optical activity, number 3, polarity, and number 4, the nutritional value. Coming to the first, based on the structure, the amino acids have been categorized into the aliphatic amino acids or the simple amino acids. In this, the R group either being non-polar or hydrophobic. Example, the glycine and the alanine. The aromatic amino acids are those which contain an aromatic ring structure, such as the phenylalanine and the tryptophan. The heterocyclic amino acids are those which, in addition to the ring, may contain the basic amino acids in them. For example, the tryptophan and the histidine. Based on the optical activity, the amino acids have been categorized into optically active and optically inactive amino acids. As you have learnt, the alpha carbon atom present in the amino acid, excepting the glycine, are asymmetric. Hence, they can exist in two different geometric arrangement or the spatial arrangement. Therefore, they are called as the isomers. Now, these isomers can exhibit the property of optical rotation. What is optical rotation? They are able to rotate the plane of light of polarization. If they are able to rotate the light or the plane of light of polarization towards the right, they are denoted as the D forms or the dextrorotatory forms and given by a positive sign or the plus sign. So what does plus sign indicate? They are optically active, number 1. Number 2, further they are able to rotate the plane of light of polarization towards the right. Therefore, they are called as the dextrorotatory. On the other hand, if they are able to rotate the light towards the left, then they are denoted as L or minus and given by the name or called by the name levorotatory. So, what does this indicate? The D and the L isomers indicate that they are optically active, have the ability to rotate the light of polarization either to the right or to the left. Why is this possible? Because they are the isomers. They can be present in two different forms or the geometric arrangement. Few amino acids like the glycine, they do not have the asymmetric carbon in them and therefore they remain optically inactive. Based on the polarity, The amino acids can either be the polar or non-polar. They can be either positively charged or negatively charged. Now, polar examples, if they are negatively charged, aspartic acid, glycine, glutamic acid, serine, and the non-polar include those which are uncharged and could be the alanine, valine, and so on. Based on the nutritional value, the amino acids could be categorized as essential amino acids. What does this mean? They are absolutely required for the normal growth and development, but cannot be synthesized by the living systems. Now, how do they obtain? It has to be obtained in the diet, in the food what we consume. So those amino acids which are required for the normal growth and development, which cannot be synthesized, have to be obtained from outside in the form of food or diet, are called as the essential amino acids. Example, the methionine and the lysine. You have the non-essential amino acids. Non-essential does not mean that they are not required for the growth of the system. They are required, but they are synthesized. They are produced within the living system, and these which can be synthesized in the body are called as the non-essential amino acids, which include the glycine and the alanine. You have the third class of the amino acids, which are called as the semi-essential amino acids. What does it mean? They are partially synthesized, but when the demand increases, the external outside supplements have to be provided. Like, for example, during pregnancy, certain amino acids, the demand of certain amino acids will not be able to meet the quantity of the essential amino acids which are synthesized in the system. So, in addition to those synthesized, you will have to procure it, you will have to supplement it from outside in the food. So, they are called as the semi-essential amino acids, like for example the arginine and the histidine. Coming to the properties of the amino acids, the optical activity, as I told you, because of the presence of the isomers and the presence of the asymmetric carbon, two different types would be obtained, and which are called as the isomers. These isomers exhibit the property of optical rotation, that is, they are able to rotate the plane of polarized light. So when in a solution, they are said to be either optically active or optically inactive. Optically active if they are able to rotate the light, the plane of polarized light towards the right, then it is dextrorotatory. If they rotate the plane of polarized light towards the left, they are said to be levorotatory. Then, amphoteric property. The amino acids are amphoteric in nature. What does it mean? Amino acid. They have a COOH group, which is acidic, the carboxylic group, which is acidic, and the amino group, NH2 group, which is basic. So therefore, they have both the characteristic features of an acid as well as the base. Therefore, it is said to be amphoteric in property. Isoelectric point. It is nothing but the pi. Isoelectric point is the pH at which the number of positive charges equals to the number of the negative charges. So at an electric field, there is no movement or either towards the anode or towards the cathode. So that is why it is said to be an isoelectric point. So why is this helpful? For the elimination or to identify
the particular amino acids which are present. It is referred to as the amino acids which are present. It is referred to as the isoelectric point. Now, zwitterions, that is, they contain both a carboxylic group as well as an amino group. Since they have both an acidic as well as the the basic in it, the basic ions in it, it is referred to as the zwitterions. Now, how does the synthesis of amino acids or the proteins take place? One amino acid is conjugated to the other amino acid which is adjacent through the formation of a peptide bond. So now, as it joins, it gives rise to a number of innumerable, indefinite linear structure of the amino acid, which is called as a peptide. And many such amino acids aggregate together with the involvement of many peptide bonds. They are called polypeptides. All these conjugate together, link together to give you a large macromolecule which is called as a protein. So the proteins can be categorized into the simple, the conjugated as well as the derived. So what are first of all the proteins? They are nothing but composed of building blocks of amino acid residues which on hydrolysis may yield just one constituent amino acid. Then it is referred to as the simple protein. Further, it could be the globulins or the fibrous proteins or the scleroproteins. Globulins, the albumin, the globulins, the histones are its examples. When they form a fibrous structure, they are also referred to as the scleroproteins, like for example in the muscles, the collagen, the elastin, and the keratin, the hair and the nails, they are composed of the keratin. They are all the fibrous proteins. You have the conjugated proteins. The conjugated proteins are nothing but proteins combined with the non-protein moiety. Further, there could be the nucleoprotein. Protein plus the nucleic acid is nucleoprotein. Glycoprotein. Protein plus the carbohydrates, glycoprotein. Lipoprotein, proteins conjugated with, attached with the lipids. So they are called as the conjugated protein. Protein part with the non-protein part. Then you have the derived proteins, derivatives of the degraded proteins which are derived from either the degradation of the simple or the conjugated proteins are called as the derived proteins. They are composed of, again, the primary derived proteins, the coagulated proteins and the metaproteins, or the secondary derived proteins such as the peptones, the polypeptones and the peptides. Coming to the structure of the proteins, they can be conveniently be divided at 4 levels of organization, namely the primary structure, which consists of the amino acids sequence in a linear chain, which are joined together, held together, attached together by the peptide bonds. You have the secondary structure. Just look at this. These are 2 different chains which are held together, joined together by the linkages, peptide linkages. Then it is referred to as the primary structure. You have the secondary structure which includes various types of local conformation in which the side chains are not involved but formed by the repetitive pattern of the hydrogen bonds between the backbone atoms. Further, there could be the alpha helix or the beta pleated sheet. The alpha helix
is nothing but the twisting of the helix in a right-handed structure. You have the beta sheet wherein the hydrogen bonding is seen between the two strands. The tertiary structure refers to the overall three-dimensional structure that is produced due to the hydrophobic interaction, or could be due to the electrostatic interactions or the hydrogen bonds, which are all non-covalent bonds. A quaternary structure denotes or refers to an interaction of one or more subunits to form a functional protein in the spatial arrangement of the subunits in a protein that consists of more than one polypeptide chain. There is another concept, what you call as the denaturation of the proteins. The proteins can be denatured by agents such as heat or ammonia, which brings about the unfolding of the polypeptide chain. Following this unfolding, there is the disorganization of the native structural protein, which may result in the loss of either the secondary or the tertiary or the quaternary structure. And this loss or the unfolding of the native protein leading to its loss of activity is what you call as the denaturation. It could also be due to the the cleaving of the non-covalent bonds, those interactions, the hydrophobic interactions or the electrostatic interaction or the hydrogen bonds, they are disrupted, disturbed, cleaved, broken down, degraded, and resulting in the change of the physical, chemical as well as the biological characteristics of the protein molecule. If the denatured protein returns to its native state, that is, after the denaturing agent is removed, after the removal of the denaturing agent, the activity of the protein can be regained, and this process is what you call as the renaturation. Denaturation, loss of activity of the proteins. Renaturation is the disrupted or the denatured protein returns back to its native state and takes up the function. Now, coming to the functions of the proteins. The proteins are vital for the growth and repair and for number of functions. The enzymatic catalysis is one such function. With the enzymes are composed of amino acids which have many roles to play in the living system. So they act as enzymes, they act as hormones, they act as antibodies. They also act as structural components such as the keratin of the hair and the nails, and important constituents of the collagen and the elastin of the bones and the muscles. They also execute their activities in the transport of oxygen and carbon dioxide by the hemoglobin, and also uptake of a lot of the ions across the cell membranes. They also play a major role in the regulation of certain physiological processes such as the blood circulation and intestinal fluids through the presence of the plasma proteins. They also act as the defense and the protection processes against infections, by means of producing the antibodies, which are nothing but the proteins. They are also acting as the storage proteins, especially the albumin and the gluten. They are the storage proteins. The actin and the myosin act as the contractile proteins, which are important for the muscle contraction. A quick recap of proteins. You have learned today about the amino acids, their importance, classification, and the properties such as the isoelectric point and the zwitterions. You are now familiar with the primary, secondary, tertiary, and the quaternary structures of proteins. You now know what a peptide bond is, and then what denaturation of proteins is, and the different functions of the proteins in the biological systems. Thank you.